using potential chemical elicitors
Yes,
to kind of, I guess, mitigate some of the negative effects of HLVd. So please tell us more about that.
yes. So chemical elicitors or synthetic elicitors, also called plant growth regulators, as the name suggests, it's a chemical that mimics plant hormones. So it's essentially giving the plants a little boost to get their defenses going. And this is not, I didn't come up with this idea. Uh, we've been using it in plant pathology and in entomology research as a way to trigger plant defenses, give them a little boost to get it activated. You could also think of it as a way to prime the plants, and it can protect against diseases and pathogens, because naturally plant hormones, or what we call phytohormones, are doing this, but they're doing it at a low level so that the plant can devote all the resources to the different functions. We want to give it this boost so it can get activated. And the reason that I was interested in this research is because cannabis and hemp can be considered organic production systems, right? Because the repertoire of insecticides and pesticides are so limited.
Right.
All we have, and so there are only a handful of products. We don't even know their efficacy. And it turns out that these chemical elicitors are actually registered for use. So that's one of the reasons we were interested in this. And so our idea was to just boost the plant's inherent immunity to better defend the viroid.
And so is this prior to infection?
No, we didn't do it prior. So we infected the plant, and a week later, so we got the plant going with the infection, and then a week later, we sprayed these elicitors, and then sprayed every week for up to 3 weeks. And then subsequently, I think at the 4th or the 5th week, we took tissue and analyzed for HLVd levels using qPCR, and then let the plants go to flowering, and then did the cannabinoid analysis. So we debated, should we do it spray first, then infect, or infect and then spray? We decided to do, and I decided to do the former, because so we got clones from infected plants, so they were already infected, ready to go. Because that's probably we figured that's probably more of the natural situation. The plants are already infected.
Excellent. And so what were the results?
Oh my goodness. So you know this already, and this is how it goes with research. It's never, yes, perfect result. Always more questions to be asked.
Trade-offs, right?
Yes, exactly. But it's great. We did 2 rounds of it, and we're actually getting ready to do a 3rd round. So the results were mixed bag, but very interesting. What we found was, yes, indeed, a certain phytohormones or chemical elicitors reduced HLVd levels. Interestingly, they also increased the biomass, the yield of the plant, which we were excited about. But go to look at the flower production and cannabinoid levels, and it was, the major ones like CBD and THC was actually reduced. I'll tell you, biomass, like almost 50% for salicylic acid. The biomass
increased 50%.
Yeah.
Compared to an infected plant?
Yeah. No, they're all infected compared to, so the control was ethanol, because that's the solvent salicylic acid dissolved in. In ethanol, the biomass per plant was 8.3 grams. And in the salicylic acid sprayed, it was like 15%, 15 grams.
Oh, wow.
Yeah. So
almost double.
Almost double. Because these phytohormones also cause the plants to cause cell division, delay senescence, and essentially giving it better overall health.
Excellent.
So I'm just picking salicylic acid. They all look pretty much the same. Some of the other ones I'm saying, brasinolides was 12.8 grams compared to 8. So a little less than 50. So let's look at cannabinoid. We can again pick salicylic acid, CBD. This is in micrograms. Okay, so THC or THCA was 1900 microgram per gram in the control in ethanol. And in SA, it was 1300 microgram per gram, 30%.
So if basically 100% increase in biomass,
Yeah,
but a 30% decrease in THC.
yes, in THCA.
Interesting.
Interesting, yeah, but not so interesting because guess what? Just after we did this, this was a 2 year study from 2022, and he graduated in fall. In fall, here comes a really nice study, and I can talk about that, showing that in fact higher levels. So our goal was to cause induction. The aim of our study, because we were kind of going blind, was we want to make sure these pathways in the plant, salicylic acid or what have you, was turned on. So we went a little high on the dosage, and then a paper came out saying higher dosage can reduce cannabinoids, whereas the lower dosage. So since that paper came out, we've got to fine tune the rates. It's not so over applying it can improve, reduce HLVd levels and biomass, but does not have the same impact for cannabinoids, which is important. So you've got to fine tune this. And so we're redoing that experiment. It's already started, and I should have some new data for CannMed.
Excellent.